The Effects of Roads and Trails on Native and Non-Native Vegetation by Michelle Hartwick, Marissa Matho, and Mariah Tengler. In the San Francisco Bay Area, the pace of development is fast, and the impacts of increased human development on natural biodiversity can be harsh. Previous research by Coffin 2007 found that roads can affect the structure and dynamics of ecosystems and have direct effects on ecosystem components such as species composition. Due to the fast-paced development in the Bay Area, particularly in environmentally and economically important regions, an assessment of the impact of roads on species composition is needed. This assessment would allow us to understand whether the addition of roads to natural habitats has led to increased vulnerability to invasion by non-native species, and any potential ecological effects this may be having on these areas. The Golden Gate National Recreation Area, or GOGA, is a national recreation area spanning Marin, San Francisco, and San Mateo counties. The GOGA is home to many sites, including such places as the Marin Headlands, Fort Mason, Muir Woods National Monument, and the Presidio of San Francisco. These sites attract nearly 16 million visitors each year, making it one of the most visited sites of the National Park Service. Our goal was to better understand how the presence of roads impacts the prevalence of native versus non-native species within managed parcels of the GOGA. Specifically, we aimed to understand how proximity to roads influences distribution of native versus non-native species. CalFlora, a nonprofit dedicated to California plants, has a wealth of observational data for many plant species. Phone applications such as iNaturalist and CalFlora's Observer Pro allow citizen scientists to record observations of plant species and locations. Using point observations downloaded from the CalFlora database, we have conducted an assessment outlining the impact of changing road developments on native versus non-native species.
Initially, roads and trails and species point observation features within the managed GOGA parcels were selected. These layers were all intersected, creating a separate layer for each, representing only features within the desired study area. From the roads and trails, a multiple ring buffer was created at 50-foot intervals from 0 to 300 feet, with the last two rings spanning 100-foot intervals from 300 to 500 feet. In this call-out portion of the map, a clustering of non-native species can be seen with close proximity to the roads. The roads and trails map was spatially joined with the native and non-native point observations, respectively. This would allow us to get a count of native and non-native species per buffer zone, from which maps displaying the respective counts of native and non-native species were produced. The species per buffer zone maps will allow us to show the count of species at a given distance, while combined map of both species types will allow us to see the larger trends in species location at distances. Shown here in this chart and table is the count of non-native and native species at incremented distance to roads and trails. From our analysis, we found high count of non-native species at close proximity to roads and trails that decreased with increasing distance. Overall, 9,658 observations of non-native species were found within 0 to 50 feet of roads and trails, compared to 1,304 observations of native species. From our analysis, we were able to establish total count of non-native and native species point observations every 50 feet from roads and trails, and determine that there is a higher prevalence of non-native species at close proximity.
Some considerations should be noted for this particular analysis. CalFlora species observation data is reported by citizen scientists and, as such, has some weaknesses. Since these observers are not highly trained, species identification may not be completely accurate. Observations are also most likely obtained while observers are actively using roads and trails, potentially making near road and trail observations overrepresented in the dataset. As an extension of this study, one could examine current GOGA land management policies and assess the restoration efforts made to protect native species. In San Francisco, San Mateo, and Marin counties, Increasing urban development has significant implications in the loss of biodiversity and local habitats. Data from our project and other ongoing research can be used to anticipate the spread of non-native species and implement policies to prevent the introduction of non-native species. Removing non-native species and planting native species in their place, as well as restricting access to particular areas, such as not allowing horses or dogs on some trails, would help mediate human disturbance. Roads and trails can have significant impacts on the immigration of non-native species into the many parks within the Golden Gate National Recreation Area. Our analyses have found that within the managed areas, the highest counts of non-native vegetation species are closest to roads and trails. These data may inform how native and non-native vegetation types are carried throughout protected areas. This assessment can also be used in future decision-making. When considering the building of or improvement of roads in specific areas, the potential spread of non-native species should be taken into account. Thank you to the GSAL at the University of San Francisco, the GIS-2 intermediate level class, and Professors Megan Danielson and Fernanda Lopez Ornelas for all their guidance. We would also like to thank CalFlora and the Golden Gate National Recreation Area for allowing the use of their data.